Hello guys, in this video I will explain part 1 section 1 of chapter 3. Let's start to talk about the non-specific defense of the body, what we mean by non-specific defense and what we mean by the infectious diseases. So the first definition that we have is infectious disease. What we mean by infectious diseases are the diseases that are caused by agents like pathogenic bacteria, viruses, fungi or protists. All of these microorganisms that have invaded the body. For example, when a type of virus, which is called rhinovirus, enters the human body, it can cause a common cold. So here we have the virus, the name of the virus, rhinovirus, which can lead to infectious disease, which is the name of the infectious disease here that we have, it's common cold. Now we have another definition here, which is pathogen. What we mean by pathogen? It's any agent that can cause disease. For example, we have different kinds of bacteria that can cause diseases, harmful disease, and we have viruses like coronavirus, we have fungi like, for example, tinea, and other type of organisms like protists. All of these microorganisms or agents can lead to diseases are named as pathogens. And the diseases that they cause are named as infectious diseases. The pathogens can be spread to humans in 5 main ways, including air, food, water, person-to-person contact, and finally through animals or the bites of animals. Here we have a table that shows some types of diseases and the pathogen that lead to these infectious diseases, and also the way of the transmission of these diseases. The first one we have the botulism. This is the name of the disease, which is caused by a pathogen named as Clostridium botulinum, which is a species of bacteria that can lead to a disease named as botulism. So here we have, this is the name of the infectious disease and the name of the agent or the pathogen that can cause the disease. The method of transmission of this disease is transmitted through contaminated and expired food. The second disease that we have is amoebic dysentery. Amoebic dysentery is another type of disease or infectious disease which is caused not by bacteria, it is caused by protists, kingdom of protists, which we have this species of this kingdom which is named as Entamoeba histolytica. So what is Entamoeba histolytica? It's the pathogen or the agent that lead to this disease which is named as amoebic dysentery. It transmitted through contaminated food and contaminated water. Then we have the third disease that we have here, it's athlete's foot, and this disease it's caused by fungi, and this type of fungi or the species of fungi that lead to athlete's foot is named as tinea. So the name of the pathogen here or the agent is tinea, and the name of the infectious disease is athlete's foot. It's transmitted through contact with contaminated surfaces and person-to-person contact. And finally we have AIDS, acquired immunodeficiency syndrome, which is caused by the pathogen which is named as HIV, human immunodeficiency virus, which is a type of virus that we will explain it in detail later. It transmitted through sexual contact, contaminated needles, and also through contact with contaminated fluids from a mother to a fetus or to infants. So pay attention here that we have botulism and amoebic dysentery. Both of them transmit through contaminated food, but here we have contaminated and expired food, while here in amoebic dysentery it transmits through contaminated food and also contaminated water. And the difference between them is that this is caused by pathogenic bacteria, while this amoebic dysentery is caused by pathogenic protists. Identified pathogen. Robert Koch, he was the German doctor and he was the first person to establish a step-by-step procedure for what? For identifying a particular pathogen that can cause infectious disease. The disease that he studied was anthrax, and the particular pathogen that he discovered which caused this disease was rod-shaped bacteria. He studied anthrax, and what we mean by anthrax is an infectious disease, and this disease of cattle that can transmit to human also. This disease is caused by what? It's caused by rod-shaped bacteria. So anthrax is not caused by virus, it's not caused by protists, instead it's caused by bacteria. And which bacteria? Rod-shaped bacteria. Then he started his experiment, and as we know from grade 10, we taught about the scientific research and how the scientists can do their scientific research. And this scientific research have many steps. The first step that the scientist should be start with is observation. Observation, for example, for something unusual, for a disease, for example, for a pathogen. And after the observation, the scientist should put the hypothesis for his experiment. And what we mean by the hypothesis, it's like a guess for the results that he expects for his experiment. After the hypothesis, the scientist should do an experiment to test his hypothesis. Then after the experiment, the scientist should put the results, the conclusion for his experiment, and finally put rule or theory. What Robert Koch did, he first observed. What the observation of Koch was? The observation of Koch was that the cattle with the illness had swarms of bacteria in their blood. So he observed that these cattle with the illness that have the disease, which is the anthrax, they had swarms of bacteria in their blood. It means they had what in their blood? They had groups of bacteria in their blood. So again, the observation of Koch was the cattle with the illness had groups of bacteria in their blood. What he hypothesized that these bacteria, which mean these bacteria, the swarms of bacteria that he finally found that rod-shaped bacteria cause anthrax. And finally, after his experiment and after his results, he concluded that isolated species of bacterium, here we have a single bacterium, rod-shaped bacterium, it caused the anthrax. Now let's go to talk about Robert Koch experiment and how he tests his hypothesis. As we said before, he observed something. He observed that cattle with the illness had swarms of bacteria in their blood. He hypothesized that this bacteria cause anthrax. Now, how he tests this hypothesis? He tests this hypothesis by doing an experiment. The experiment of Koch started by what? Started by that Koch isolate rod-shaped bacteria. For example, he take the sample, for example, from the cow, and after that, when he take these samples, he found different kinds of bacteria. He isolate only rod-shaped bacteria. For example, let's say he have swarms of bacteria, rod-shaped, bacilli, cocci, and then finally he isolate only the rod-shaped bacteria from a cow with anthrax. Then he grew colonies of the bacteria. We will talk about it now in details, how they grew these colonies. To be ensured, he isolated a single species. So why he grew colonies of the bacteria? To ensure that he was isolated only a single species of bacteria. Then he injected the healthy cows with these isolated bacteria, and the cow had got developed anthrax. Koch found that the blood of these cows contained the same rod-shaped bacteria as the first cow. This is a result. Furthermore, he found that healthy cows that he had not injected lack this type of bacteria. This is another result. Finally, he put his rules, and the rules of Koch was named as Koch Postulates. And they are the rules that used for determining the cause of diseases. Scientists have used Koch postulate to identify thousands of pathogens till nowadays. Let's go to talk about the Koch postulates, or the rules of Koch that used till nowadays in order to identify the pathogen that can cause infectious diseases. In the first step, what he did is that the pathogen must be present in an animal that has the disease and absent in healthy animal. So he take two animals, or we can say two groups of animal, group of healthy animal and group of infected animal. The pathogen must be present in the animal that have the disease and absent from the healthy animal. This is step one of Koch postulate. The pathogen must be present in animal that has the disease and absent in healthy animal. In the second step, what he did, the pathogen must be isolated from the sick animal and grown in a laboratory. What do you mean by that? He take the pathogen from the infected animal and he grown it in laboratory by using petri dish. What you mean by petri dish are these dishes that we explained them in grade 11. We said are those type of dish that we put agar-agar in it in order to use it for the growing of microorganisms. So when he put the bacteria here, the bacteria grown here and form colonies. Why he grown the bacteria and form colonies or thousands or millions of these bacteria? In order to be ensured that he isolate only a single species of bacteria. For his experiment was the rod-shaped bacteria. And the third step, when the isolated pathogen is injected into a healthy animal, the animal must develop the disease. So when we have isolated pathogen that we had from here, he takes sample from here, from this isolated pathogen, and he put it here. He inject this pathogen into the animal, into a healthy animal, and the animal must develop the disease. So this healthy animal must develop the same disease that found in the first animal. For example, for Koch experiment was the disease which is named as anthrax, and this sample here is rod-shaped bacteria. In the last step, the pathogen should be taken from the second animal and grown in the laboratory. The pathogen cultured from the second animal should be the same as the pathogen that cultured from the first animal. So what he did, then again he take the pathogen from this animal and he grown it in laboratory by same way, by using petri dish and form colonies. And when he cultured it, he found that this pathogen is the same pathogen that he found it here. So the pathogen is the rod-shaped bacteria, and the disease that the animal is infected by is anthrax. Here is another figure for the experiment, and this is Robert Koch, and Robert Koch rules are named as Koch postulates. Koch postulates include these 4 steps that you should memorize in order, please. And we said that in the first step we have the animal. The pathogen should be present in the infected animal and absent from the healthy animal. And in the second step, what he did, he isolated the pathogen from the infected animal. You can see here that the healthy animal does not have the pathogen, while the infected animal have the pathogen, and he grow colonies of it to be ensured that he isolate a single pathogen. After that, he injected this pathogen again into another healthy animal, and after that what he did, he cultured it, he grew it in colonies again, and he cultured it. When he cultured it, he found the same pathogen that he found it in the first animal, who's infected animal. We have 2 questions here about this subject. First, an agent that causes disease. Is it A, tinea, B, anthrax, C, botulism, or D, all of them? First, we have botulism is the name of disease. Anthrax is a name of disease. So they are not agent that cause disease. They are not pathogen. Only we have tinea. It's the agent here, which is the pathogen. Name of pathogen, tinea, which is a type of fungi that cause athlete's foot. Second, Robert Koch was the first person to study rod-shaped bacteria, identify pathogen that cause infectious disease, and discovered anthrax or all of them. He only identified pathogen that cause infectious disease. He not studied rod-shaped bacteria, because rod-shaped bacteria is studied before this scientist, and he not discovered the disease which is named as anthrax. He only identified the pathogen that can cause infectious disease. He identified that this disease which is named as anthrax is caused by a pathogen with rod-shaped bacteria. In the next lesson, we will start with the body's defense system, that we can divide it into 2 main parts. Non-specific defense system, that we will explain it in detail in this section, section 1, and in section 2 we will talk about the defense system that is named as specific defense. We can say it's the immune system, and the non-specific defense include mucous membrane, skin, inflammatory response, acidity of the stomach, temperature response, with the proteins that involve both complement protein system and interferon, while the specific defense immune system include both cell-mediated immune response, T cells, and humoral immune response, B cells. Thanks for watching the video. See you in the next video.